Think about this for a second. Every single time one of your cells divides, it has to perfectly copy a book that's over 3 billion letters long, and it does it in just a few hours. That book, of course, is your DNA, and the copying process is called replication. So how in the world does it pull off this incredible feat with almost zero errors? Well, today, we're going to unravel the amazing molecular machinery that makes it all happen.
And it all starts right here, with probably the most famous shape in all of science, the DNA double helix. When Watson and Crick figured this out, they realized its very structure held a secret, a massive clue about how it could duplicate itself. So that's our central question. How does this beautiful, twisted ladder create an exact copy of itself?
You know, the first clue to solving this whole mystery didn't actually come from a new experiment. Nope, it came from the brilliant minds that discovered the structure in the first place. They just looked at their own model and had this incredible aha moment.
I love this quote. It's got to be one of the biggest understatements in science history. What Watson and Crick were saying here, in this very subtle way, was absolutely revolutionary. See, because A always pairs with T and G always pairs with C, the two strands are basically mirror images of each other. If you have one strand, you automatically know exactly what the other one looks like. It's like a built-in blueprint for its own duplication.
So this idea got a name: semi-conservative replication. The theory was beautifully simple. The original or parental DNA molecule unwinds, and each of those two old strands serves as a template to build a brand new partner. And the result? You get two new DNA molecules, but each one is a hybrid. It's half old and half new. So here's the thing. It was a brilliant idea, a really elegant theory on paper, but proving that this is what's actually happening inside the chaotic, messy world of a living cell, that's a whole different ballgame. I mean, how could you possibly tell which strands were old and which ones were new? And that brings us to the definitive proof. It was a landmark experiment, just so clever, done back in 1958 by two scientists, Matthew Meselson and Franklin Stahl. They came up with a genius way to make the old and the new DNA strands visibly different from each other. Okay, so here's their setup. It's brilliant. They used two different versions, or isotopes, of nitrogen. Just think of one as a slightly heavier version of the other. First, they grew bacteria in a broth full of this heavy nitrogen, called 15N. Since nitrogen is a key part of DNA, all the bacteria's DNA became heavy. Then they moved the bacteria into a new medium with normal, light, 14N, and just let them divide. By taking samples over time, they could literally weigh the DNA and see what happened to those original heavy strands. And the results, oh, they were just beautiful. You can see them here. When they spun the DNA samples in a centrifuge, the original all-heavy DNA formed a band low down in the tube. But after one generation, just 20 minutes, there was only one band, and it was sitting right in the middle. It was a perfect hybrid of heavy and light one. This was the smoking gun. It proved each new DNA molecule had one old, heavy strand and one new, light one. Then, after a second generation, they saw two bands, one hybrid band and one purely light one. That's exactly what you'd predict if the semi-conservative model was right. The evidence was just crystal clear. The experiment proved, without a doubt, that each new DNA molecule saves one of the original strands while building one new one. The hypothesis was confirmed. A huge piece of the puzzle of life had just been solved. So the Meselson-Stahl experiment gave us the what? A beautiful confirmation of the semi-conservative model. But it also opened up this massive new question: how? What kind of incredible molecular machinery could possibly pull this off with such amazing speed and accuracy? Let's start with speed, because this is going to blow your mind. In a bacterium like E. coli, this replication machine adds about 2,000 DNA letters or base pairs every single second. To put that in perspective, that's like typing this entire script in less than a second. I mean, to work that fast and not make a ton of mistakes, you need a whole team of highly specialized molecular machines we call enzymes. So let's meet the star of the show, DNA polymerase. This is the master builder enzyme. Its job is to cruise along the unwound, single-stranded DNA template, read it, and grab the correct matching nucleotide to build the new strand. It's the catalyst for the whole operation. Now, the action doesn't happen all over the DNA at once. First, an enzyme called helicase comes in and unzips the double helix. This creates a Y-shaped structure that we call the replication fork. This is the active construction zone, right? It's where the old strands are exposed and the new strands are actively being built. But hang on. Here's a major complication. The two strands of DNA are anti-parallel. That just means they run in opposite directions, like a two-lane highway. And our master builder DNA polymerase is a one-way machine. It can only build a new strand in one specific direction. This creates a really fascinating puzzle at the replication fork. How do you copy both strands at the same time?
The cell's solution to this direction problem is it's so clever and elegant. It actually copies the two strands using two slightly different strategies. And this gives us what we call a leading strand and a lagging strand. To really get this, we have to look at a little bit of chemistry. The two ends of a DNA strand are different, and scientists label them 5' and 3'. This is based on the carbon atoms in the DNA's sugar backbone. And here is the crucial rule. DNA polymerase can only add new bases to the 3' end. Think of it like a car on a one-way street. It can only travel in the 5' to 3' direction. This rule is absolute, and it's what creates the puzzle at the fork. So because of this one-way rule, the cell has to use two different game plans. For one of the template strands, we call this the leading strand, the polymerase can just move smoothly along right towards the unwinding fork, building one long continuous new strand. Super easy. But on the other template, the lagging strand, it's pointed in the wrong direction. So the polymerase has to work backwards. It builds a series of short, disconnected pieces away from the fork.
Of course, you can't have a new DNA strand that's full of gaps. So a final enzyme has to come in and do some cleanup on that lagging strand. This enzyme is called DNA ligase, and its job is basically to act like molecular glue. It goes along and stitches all those small fragments together into one continuous, complete DNA strand. Okay, wow. We've gone from a hypothesis to the experimental proof, all the way down into the complex molecular machines doing the work. So let's bring it all together and just recap the key takeaways from this incredible process. So to sum it all up, replication is semi-conservative. Each new DNA is half old, half new. The master builder is DNA polymerase, which is ridiculously fast, but can only work in one direction, 5' to 3'. This one-way rule forces the cell to make one strand continuously. That's the leading strand, and the other one in little pieces, the lagging strand, which then gets stitched together by our molecular glue, DNA ligase. This whole intricate system ensures a near-perfect copy of life's blueprint every single time. It's the absolute foundation of growth, repair, and reproduction. But nearly perfect isn't always perfect, and that leaves us with one final, fascinating question. What are the consequences when this incredibly precise copying process makes mistakes?